Okay, so this is our first exam review session for microbiology. We're just going to go through the PowerPoint that I sent out earlier. Essentially, this test will be majority on, again, weeks 1 and 2. I had mentioned previously in week 3 that molecular information, that was more just information, background stuff. I don't really test on that. So again, the majority of our testing items are going to come out of week 1 and 2 here. So that's what we will review. A little bit on the test itself. I always like to give some insight for the test. Basically, it is worth 57 points. It is a combination of multiple choice, matching, and short answer questions. So there's a good amount of short answer questions in there, a few matching, and then a bunch of multiple choice. So just kind of give you a perspective on what to expect for when you go to take the test. So, all right, now that we've talked about that, let me share my screen so we can jump into the PowerPoint. All right, so there is the PowerPoint that I sent out earlier, and we are just going to go through this. All right, so jumping back to week 1 items, we didn't really lecture through week 1, but I felt like they were pretty simplistic in nature to remember. What we should remember out of week 1 with the taxonomy part is taxonomy's purpose is always to classify, name, and identify organisms. It allows all scientists, people in general, just to use the same naming. So if I'm talking about Staph aureus, we're all talking about the same Staph aureus organism. So there's always a genus. The genus is going to be the first part of the name. So in this example, Staphylococcus is the genus name. And then after the genus will become the species name. So the species, so the genus is kind of like, if you think of it this way, your family name, like your last name. It means that you're all roughly relative and very similar to each other. And then when you go to the species name, that's even more similar, like it's whittling it down to even lesser people having in common. So that's kind of what it is. So it's genus and then species. So again, these are grouped up because they share the same physiologic and genetic features. All right, so with that being said, there is a proper way to always write the genus and species name. The genus is always capitalized, and then the species is always lowercase, and you would italicize both parts. So if we were to look at this question, which one is correct, we again want a capitalized genus, a lowercase species, and everything italicized. So in this case, letter A would be the correct format in which you would write out the bacterial name. I don't really look for this a lot in our assignments for like lab reports and things, but it will become important whenever you write like a research paper. I know in this class we have 2 case studies that are things that you write out. In the case studies, I will be looking for you to write it out correctly with capital genus, lowercase species, and everything italicized. So do keep that in mind. That is the correct way to see the organism names written. All right, within week 1, there also is discussed on what are phenotypic characteristics. Phenotypic characteristics, just like people, are things that you can see. So if you ever learn genetics, you should have learned that people have phenotypic characteristics. It would be your blue eyes, brown hair, you know, things that you can actually see. For an organism, we have the same thing. In this case, it would be their colony morphology. So what do they look like on the agar plate when they grow? What color is that colony? Does it have a certain shape to it? Does it have hemolysis? Those are things that we could take into account. You know, the gram stain characteristics, is it gram-pos or gram-neg? What is the shape on the gram stain? So that is all considered phenotypic characteristics. Along with that is maybe potentially how it grows in oxygen, if it's an anaerobe or an aerobe, and then any resistance it might show to certain antibiotics. So things that we can visually see. There was also a lot of talk about genetics in week 1. Usually I very, very lightly skim over bacterial genetics. To me, at the level of an associate degree, we don't delve too deep into genetics. That would be if you guys go on to pursue a bachelor's degree, you're going to get a lot of more science-based general courses. And in those general science-based courses, they'll talk a lot more about genetics. So that's where they'll delve in there. But as far as what we need to know, some of the things they ask are these terms right here, what a plasmid is. A plasmid are miniature chromosomes. So they do consist of genes in a circular structure. So they're kind of considered mini chromosomes. A gene is your actual DNA sequence that will code for a product. And then a genome is all the genes put together in an organism. All right, we can achieve genetic diversity through the three following ways. This just basically means how can something change and adapt to situations and survive? So we've heard of organisms and bacteria becoming mutated, and that happens because they're trying to survive their environment just like we are. So if they need to figure out how to become resistant to different antibiotics, they will do so. They'll mutate. They will change to make sure they'll survive. They can also have genetic recombination and genetic exchange with other organisms. All of this allows them to adapt to their environment and continue their survival. So that's what we consider genetic diversity. So they can occur through one of those three ways. Within week one was a lot of safety kind of things. There were some terms in there that I think you had on an assignment or a lab report that you answered, which was what is a disinfectant, what is a sterilization process. So a disinfectant will kill microbes, but not necessarily everything. It's things that we use commonly would be our bleach solution that we use on the countertops every day, whereas sterilization is ensuring everything gets killed. So a good example here would be an autoclave system. If you ever go to the dentist, they autoclave all those dental tools to ensure that every microbe that got picked up in somebody's mouth is killed off and it's perfectly clean for the next patient. So the way that an autoclave, for example, works is it's using a steam heat. There are dry heat systems, but autoclave is actually working off a moist steam heat, usually around 30 to 60 minutes at a high temperature and high pressure rate. That is something I would remember. I do thoroughly mention that there for a reason, so just be sure to remember that. And then you also had an assignment in week one where you discussed the 4 levels of biosafety. There's technically 3 biosafety cabinets and 4 biosafety levels. I know it's a bit confusing, but level 1 basically means hardly any harm will come to us with those organisms, so they're not much safety concern there. Level 4, extreme concern, that would be your Ebola, your smallpox. The one that we most commonly use in hospital labs, which you guys had a question on this, was level 2. So level 2 is the most common one you'll see in hospital labs. Again, which level offers maximum protection? That would be your level 4. All right, some terms that you also answered within week 1 assignment. Normal flora are just bacteria that live in and on us. They're inside of us. They're part of us. They're there to actually serve a purpose. They're there as part of our immune system, if you will. So they kind of help make sure that other organisms don't try to come and take up residence. Opportunists are those that take advantage, take opportunity of a situation. So if your immune system is down and you're kind of wiping out your normal flora, that's their chance to be like, oh my goodness, we can flourish, we can multiply, we can cause our disease. So that's opportunists. Colonizers, the key words on colonizers are persistent survival. They're not necessarily normal flora. They're not necessarily always found on us, but sometimes they can persistently survive on the body. And then a pathogen is anything that will cause a disease or infection. Nosocomial means it's hospital acquired, so that is an infection you got within the hospital. For example, if you were admitted to the hospital because, say, you had a heart attack and you're in the hospital with that heart attack, and all of a sudden the next day or a couple days you have a UTI as well. You didn't come in with the UTI, you acquired it upon being there. That would be a nosocomial infection. And mode of transmission. When we start discussing bacteria, which will be week 5, we will discuss sometimes how we get the bacteria. What is the mode of transmission? How does it come over to a patient? So there's a lot of different ways you can get bacteria. Sometimes this involves what we call a vector or a fomite. So a vector is anything living that transmits the organism. So it could be something living. It would be a mosquito, a tick, a fly, a gnat, things like that. Something that can carry that organism and bite you and then transport the organism into you. Whereas a fomite is a non-living item. That would be a surface of the counter, a doorknob, your purse, a cell phone, the toilet handle, anything that somebody sneezed on and you picked up whatever they had or something like that. So again, vector living, fomite non-living. All right, when we do get into discussing bacteria, we will eventually learn that they have different growth requirements. Things that are considered aerobes mean they need oxygen. Anaerobes strictly require no oxygen. Capnophiles, you did define this on your week 1 assignment, so I go back and look at that. Capnophiles means that they want an increased amount of CO2. Capno always refers to CO2, carbon dioxide. Capno is like the medical terminology for carbon dioxide. Phile means loves. It loves itself in carbon dioxide. So the word itself tells you that they're going to want an increased CO2 environment. Now, there's another term called microaerophiles, and you can tell by the word micro for less, aero for air. They want less oxygen in order to grow. Now, there is a fine difference between capnophiles and microaerophiles. There are percentages that are different. And you guys did a good job on week 1 of defining those. I would definitely know those percentage numbers. So with capnophiles, they'll increase the CO2, but the oxygen is still, quite a bit higher than it is in microaerophiles. In microaerophiles, the oxygen is less. So go back, relook at what those percentage numbers are on each of those. And again, you guys did this in the week 1 assignment. All right, I don't know if I put this in somewhere. I can't remember now if it's in the week 1 PowerPoint. At some point in the week 1 PowerPoint, there's somewhere when we went through all these bacterial growth requirements, there's always a special mention to Campylobacter jejuni, which is C. jejuni here, Campylobacter. Most bacteria want to grow at a body temperature in an incubator. So body temp is 35 to 37 degrees Celsius. So in the lab, all of our incubators are typically set to 35 to 37 degrees Celsius range because we know that that's what bacteria like and they'll really thrive in that warmth. Whereas there is a couple of bacteria that are unique, and Campylobacter jejuni is one of them. This one wants it even warmer. So this actually wants a 42-degree environment. So I do ask that you remember that. I know we haven't learned this bacteria yet, but because we learned growth requirements, we threw that in at this point. So do remember Campylobacter jejuni wanting a 42-degree Celsius environment. It wants it warmer. So usually a lot of labs will have an extra incubator set to that temperature for Campylobacter screening. Okay, so going into week 2 items. So week 2, we did have a lecture on. If you feel weak on any material in week 2, be sure to go back, listen to that lecture recording to help you go back through the PowerPoints. So we discussed a lot of staining process within the, I think it was chapter, I don't want to say chapter number because now I'm going to be wrong, but it was within the microscopic PowerPoint. So first up was gram stain, which I think you guys have now started playing with these, which is great. So we have discussed there's 4 reagents, of course, in the gram stain, and you need to know all 4. We have your crystal violet, which is your primary stain. Iodine, which is called the mordant. It helps bind the crystal violet into the cell wall. You have your decolorizer step, and then you have safranin, which is your counter stain. So those are your 4 reagents. Of course, you've been looking at gram stains under the microscope. You should all now know that gram positives are a dark purple color, almost look blue. They're so dark sometimes. And then gram negatives, by the way, I use equal signs for negative. If that ever confuses you, that equal is a negative. But gram negatives are that light pink or red color. So we asked this on one assignment, but again, let's re-ask. It's an important question. At what step is the first time you can distinguish between a gram pos from a gram-neg, and again, that would be your decolorizer step. So the decolorizer step is the first time you can start to tell apart a gram-pos from a gram-negative. And then knowing the difference, why are gram-positives gram-pos and why are gram-negatives not? So again, the whole thing is with that layer of peptidoglycan in the cell wall, gram-positives have a thicker peptidoglycan layer. They also have teichoic acid cross-links. All that together allows them to really embed that crystal violet purple stain into the cell wall, and it cannot be rinsed out, washed out, decolorized. So they will stay that purple color. Whereas gram-negatives have a thinner peptidoglycan layer. They also have an outer membrane and stuff, but they have a much thinner peptidoglycan layer, so they easily have that crystal violet wash out upon decolorization. And then when you do the safranin, they'll keep that red safranin color instead. The other big staining process that we learned in that week was acid-fast stain. There are a few techniques here, but overall, the two techniques use the same reagents. We have carbofuchsin as the primary stain, then we have an acid decolorizer, and then we have methylene blue for our counter stain. So anything that's considered acid-fast positive is going to keep that carbofuchsin red color. Anything that's acid-fast negative will keep that methylene blue color. So again, two methods, Ziehl-Neelsen and Kenyon. You guys answered some questions on this in week 2. Ziehl-Neelsen uses a heat boiling technique. So basically when they put on the carbofuchsin, then they boil it to help embed it into the wall of the bacteria. Whereas Kenyon replaced that. They took away the boiling need and instead used a higher concentration of phenol in their reagents, so they didn't have to worry about boiling it. So those are the two methods. So Ziehl-Neelsen is the hot method, Kenyon is the cold method. Overall, what an acid-fast stain is for is, again, to target the mycolic acids in the cell wall. These bacteria cell walls are much different than normal, so they won't gram stain. You can't use a gram stain on them, but instead you can use these acid-fast because it's targeting those mycolic acids. One group of bacteria that we've discussed that are hugely acid-fast positive are the mycobacteria group. And then within that chapter was also mentioned of 3 fluorescent dyes, what we call fluorochromes, and we just need to remember what each one of these binds to. Acridine orange, remember, binds to nucleic acids. Oramine rhodamine binds to mycolic acids, and chalcofluor white binds to the cell walls of fungus. So again, acridine orange binding to nucleic acids, oramine rhodamine binding to mycolic acids, and chalcofluor white binding to the cell walls of fungus. And then we got into the next chapter within week 2, and that was our augurs. That's a big chapter, and there's going to be a lot of big questions on the augurs in this exam, just to warn you. So first and foremost, we have to know the 4 categories and the definitions of the 4 categories. So supportive, and I'm just going to define these in my own words. You can look up the actual definitions in the textbook. But supportive basically means it's going to support the growth of almost anything. Almost anything will grow in that auger. The things that don't grow are typically the fastidious or picky organisms. Enrichment will add extra nutrients, extra items, enhance that auger plate so that those picky or fastidious organisms will grow. Selective will have just one group of bacteria growing and inhibit others from growing. And differential will kind of subcategorize those bacteria on other characteristics. So those bacteria that did grow, they'll use other characteristics to help kind of categorize them further. All right, so here are the augurs that I do ask that you know. I need you to know the categories that they fit into, what their purpose is, and then anything extra on each one. So I will go through this with you. I want to type as I do it, so give me a second. I'm going to get out of slideshow mode so that way I can type. And let me go back to that slide. And I'm going to type in this notes section. So the first one listed was blood augur. All right, so blood augur again is supportive and differential. It basically will grow almost anything. It just doesn't grow the fastidious. And the reason it is differential is because of the hemolysis patterns. So let's remember, beta hemolysis is a complete clearing. That's the one that kind of looks like a halo. I hope some of you guys have been able to see a few of these. If not yet, you will soon. Alpha hemolysis is a partial clearing. That's the one that kind of has that green hue to it. And then gamma is nothing. It has no hemolysis. So that's everything you need to know about a blood agar plate. So supportive and differential pretty much grows everything, and then remembering what each of those hemolysis patterns are. The next agar was chocolate agar. This is supportive and enrichment. And by the way, you guys all had that study guide in week 2 on this, and I tried to make sure when I went and corrected those, I ensured that each of you had the correct categories on there, and if you didn't, I corrected it. I put a comment. So really look at that week 2 study guide. Make flashcards out of it. Do whatever you need to do to remember these. All right, so chocolate was supportive and enrichment. This grows everything, and it plus the fastidious because it is enriched. So it's been enriched to make sure the fastidious also grow. 2 examples of fastidious bacteria, there's many, but 2 common examples that we talk about a lot are Haemophilus and Neisseria. I can't type right now. There we go. So those are 2 examples of fastidious picky organisms. All right, the next one was CNA. CNA is selective only. This is growing just gram-positive. So it has an antibiotic in it called colistin that will help inhibit gram-negative. So it only is going to grow the gram-positive, so selective only. PEA, same thing, selective only, growing gram-positives. MacConkey agar is selective and differential. So it is selective because it grows gram-negatives, and it's differential based on fermentation characteristics. So in this one, if we have what we call a lactose fermenter, LF for short, that would be that purple color on the colony. If it was a non-lactose fermenter, NLF, non-lactose fermenter, it would be clear or colorless colonies. All right, then we have hectowin enteric agar, which is HE. This is selective and differential. So again, it is selective because it's growing gram-negative, specifically screening for Shigella and Salmonella is its primary purpose. So it will grow different gram-negatives, but it's really screening for those Shigella and Salmonella colonies. All right, it is differential because of, again, fermentation characteristics. So Salmonella and Shigella are both known non-fermenters. So when they grow on hectowin enteric agar, they will be blue-green colonies. Remember, Salmonella also can produce H2S, which is hydrogen sulfide. That will give it a black precipitated center to it. I spelled that wrong. Black precipitate. Yeah, there we go. All right, so that should cover HE agar. The nice thing is XLD agar, same purpose. So it's selective and differential. So again, it's growing gram-negatives, specifically looking for Shigella and Salmonella. And then again, it's differential based on fermentation. So in this case, Salmonella and Shigella, when they grow, since they are non-fermenters, they will be colorless colonies. And then again, Salmonella will still have that black center to it because they can produce that H2S. All right, thioglycolate broth, which is a liquid media, is supportive and enrichment. So it will allow, it grows pretty much everything. This is the one that can sometimes have the unique growth patterns depending on what's growing in there. So if there were cloudy spheres, or you can say balls, whatever you want to say, circles, you know, it doesn't matter. Those tend to indicate you might have more gram-positive cocci growing. If it is diffuse or cloudy throughout the entire broth, then it's more gram-negatives growing. If it is cloudy or turbid at the top of the broth only, that means you have a strict aerobe. That is an organism that has to have oxygen, so it's up at the top trying to get that oxygen. And then the opposite, if it was cloudy at the bottom only, that is an anaerobe. It is trying to get as far away from that oxygen as possible so it can grow. So keep in mind those different growth patterns that occur in the thioglycolate broth. All right, the next two are very similar. We have Thayer-Martin, which is selective and enrichment. It is selective because it's only growing Neisseria, and that is also why it's enriched. As we mentioned up here, Neisseria is a fastidious, picky organism, so it has to have an enriched agar to grow anyway. So it's selective to only grow that, and it's enriched because that Neisseria is picky. Now, in order to make sure that only Neisseria grows and absolutely nothing else, there are 4 antibiotics in this. So I am actually going to flip to the next slide right here because I have them listed out. So you can see these antibiotics are listed for Thayer-Martin. So it has vancomycin, which you need to remember inhibits gram-positives from growing. It has colistin, which inhibits gram-negatives. It has trimethoprim to inhibit Proteus, which is just an organism that can easily take over a plate. And then it has nystatin to inhibit yeast from growing. So you do need to remember those 4 antibiotics are found in the Thayer-Martin plate and what each one inhibits in order so that only the Neisseria species comes through. Now the other agar that was last on the list was Martin Lewis. It's very similar to Thayer-Martin. Again, it is, if we go back over here to my thing, again, Martin Lewis is, here I'll write it out, is again selective and enrichment because it is only looking for Neisseria. So it as well has 4 antibiotics in it. So if we go to that next slide again, let me bring this back down. The only difference between the two is within Martin Lewis is it put in higher amounts of vancomycin, and then it decided to use mycin to inhibit yeast instead of nystatin. They just felt that it probably did a better job or whatever it might be. So that's the difference between Thayer-Martin and Martin Lewis. So please remember that. All right. So that is kind of everything for weeks 1 and 2. I would say the biggest thing to remember and memorize, period, is going to be all those agars, all the purposes, the categories, those kind of things. So really dig into the agars, dig into the stains. You know, there's definitely things from week 1, but, you know, week 2 material seems to be the stuff that takes longer to remember and get down. So if you guys know all of that, you guys should be golden. You should do really, really well. So I'm excited for you guys to have the exam and see everything. So if you do have any questions between now and then, let me know. But otherwise, please make sure you get your appointment set up with your proctor on campus. And I wish you guys the best of luck. Thank you.